I already saw two of the speakers. The second and third I didn't see. Are you here? Well, hopefully you'll come here. Otherwise, the man who was in charge of the sound told me play the sax, so we can do that. So it's a pleasure for me to introduce the first speaker, Johannes Soeding, who will show us a BLAST-like thing that detects twice as many homologs, and I see that all of you are aware of the importance of that. So with no further ado, please, Joachim.
Thank you, Nir. This talk is not only going to be about a method that is better than BLAST, but I want to show you that, in fact, it's a new way of thinking about protein evolution, about point mutations, because we're bringing the context, the sequence context of residues into the equation, and I think that is a very powerful method, not only for improving BLAST, but also basically everywhere where you're concerned with point mutations and modeling sequence evolution, so multiple sequence alignments and so on and so on. Before I start, this is based on a paper that has been published in PNAS in February or March this year, and the first author is Andreas Biegert, my first PhD student in Munich. So before we start, I want to give you remind you of the importance of sequence searches for biology, and the best, I think, to illustrate this is the number of BLAST jobs that is run at NCBI alone. It was 400,000 a few years ago per day, and another number that is interesting is the number of citations of the BLAST and PSI-BLAST papers dating from 1990 and 97, which have been cited over 45,000 times. And I think they rank still among the 20 most cited papers of the last 2 decades in all of science, of course. And the reason why BLAST and PSI-BLAST are so tremendously popular is because they offer the best balance of sensitivity and speed. The reason why sequence searches are also fundamental for bioinformatics, not only for biology, is because most of the things that we do with sequences, we actually do with sequence alignments. And the reason is that the properties that we want to predict from a sequence are mostly often conserved over the homologs. So it pays really to look at the sequence alignment because the properties will be the same for most of the sequences in the sequence alignment. And obvious examples are secondary structure prediction or other 2D predictions like solvent accessibility, coordination numbers, and so on. There are many of them. Transmembrane helices are predicted from alignments. Homology modeling relies very heavily on finding templates with profile searches or even profile searches. Then phylogeny obviously requires alignments, function prediction, domain boundary prediction, protein evolution, internal repeat detection and prediction of protein interaction surfaces, functional residues, and so on and so on. So how does sequence alignment work? Well, we all know basically all the algorithms maximize a score, which is the sum of the amino acid similarities in sequence searching, minus some gap penalties. So if we color code the amino acid similarities here in red and blue, we're just trying to find the alignment that maximizes the sum of these scores here. Now, this method really makes use of pairs of amino acids to calculate the similarity. And in order to motivate to you how we use sequence context, I want to show you that, in fact, sequence alignment can be understood as a special case of profile sequence alignment, where the profile is generated from a single sequence, it doesn't matter which one of the two, by using a substitution matrix. And the way this works is, if you look at the score in the substitution matrices, all well-performing substitution matrices are in fact log odds scores, where you have a model probability and a null model probability, and the model probability is for the score of amino acid X with Y. The model probability is the probability that amino acid X will mutate into amino acid Y within a certain time that is inherent to this substitution matrix. And if you take now this, and I've depicted these probabilities for the different amino acids here, A, C, D, and so on, for Blossom 62 matrix as an example. So if you now take a sequence and you substitute every residue with this profile column here that corresponds to the residue, you'll get a profile. And from this simple equation down here, it follows very easily that the profile sequence score that you get then is exactly equivalent to the sequence scoring using this same substitution matrix. So you could use profile sequence scoring for doing the same thing that BLAST does, and in fact PSI-BLAST can be jump-started with a profile and will then give you the same results as BLAST. Context specificity is not new. It has been investigated since the 90s, and the idea was that if you have a sequence and you know the structure of the sequence, you can learn better substitution matrices. And the way this works is that you can classify your structural contexts by, for example, secondary structure state, coil, extended sheet, or helix, and, for example, by solvent accessibility, exposed or buried. And then you just learn these, let's say in this case, 6 substitution matrices, and you will use the one that is that corresponds to your states there. And people have used this with quite some success, in particular for protein structure prediction. So this has been proposed first by Overington and Blundell, and then used for structure prediction quite successfully by Rice and Eilenberg. But the big drawback of this method, obviously, is that you need a structure. And for most of the proteins, we don't have structures. So a decisive step was taken by Huang and Bistroff, who used their ISITES library that contains, in fact, an HMM with 281 states that are determined from the sequence context. And they trained 281 sequence-dependent substitution matrices for each of these states. And they could show that they were really able to improve alignment quality for sequence-sequence alignment by this method. So the way we approach this problem is a bit different. We just take sequence context directly. That means we take the 6 neighbors to the left and to the right of each residue. We call this the sequence context. And take this example. This is a part of a zinc finger protein. And you see here, these 2 cysteines are in fact the ones that coordinate the zinc ion, whereas this cysteine here is not conserved and doesn't look like a typical conserved cysteine which would appear in zinc finger. So if you look at the multiple alignment, indeed, you find that this cysteine is conserved, whereas this one is just a small amino acid, like many of others that appear in this column. Now, if you had an algorithm or a method that could automatically find the similarity of this context to zinc fingers, it would be able to predict that this cysteine is highly conserved, whereas this cysteine doesn't really fit any kind of conserved cysteine in the context library that it would use. So this is the idea of our context method. It works by sliding a window over this sequence. So we have a residue here, C, for which we want to predict the mutation probabilities, the profile, sequence profile. And this window is compared with a pre-calculated library of context profiles. And each of these 4,000 context profiles gets a certain probability for generating the sequence. And then we just mix together the profile for these columns, for the cysteine, from the contributions of these context profiles. So we end up with a context-specific profile that we can then feed into PSI-BLAST by jump-starting PSI-BLAST with it. I should mention that PSI-BLAST runs with the same speed as BLAST, so we don't have any speed disadvantage there. The way we generate these context profiles is fairly straightforward. The key is we have to use a lot of data. We want to learn everything and we don't want to have biases. So we take the whole NR database, cluster it down to 30%, take the alignments that have a certain minimum diversity, start a BLAST search with each of them to get a bit more diversity into the alignments, And then we take these alignments, convert them into sequence profiles, and chop them up into these 13 residue windows, possibly overlapping, and then we just sample 1 million windows out of this, these profiles. And these 1 million windows are our training profiles. So they contain everything, disordered regions, membrane, helices, and so on and so on. And then we train with an EM algorithm 4,000 profiles, also of length 13 positions. We maximize the probability that these 4,000 profiles can generate these 1 million training profiles. So that's how we end up with our context library. And here are 8 typical examples of what kinds of profiles we find there. So we have amphipathic helices, not only one kind but many kinds, zinc fingers in all the different registers. This is a typical transmembrane alpha helix, coils with a typical hydrophobicity pattern, exposed beta sheets, GD box turns, for example, disordered regions, structural repeat proteins, and so on. To give you a bit of a feeling of where the differences are between the profiles generated by this simple substitution matrix method and the context-specific method, I've shown you here 2 examples. One is a disordered region, an activation domain from human transcription factor, SOX9. It has a lot of prolines to keep it disordered, probably. And you see that this substitution matrix method just assigns the standard proline column to each of these positions, whereas the context-specific profile has learned that the prolines themselves are not really that important where they are. There just need to be enough of them. So it puts less probability into the proline columns, but puts more next to them. If you look at this structured protein, you see that the cysteines here, which coordinate again a zinc ion, are less conserved in the substitution matrix profile than in the context profile, because there's some context that tells it that this needs to be conserved. Whereas, for example, the proline here, which is just a standard proline, it uses the same one here and here, the substitution matrix. The proline is higher conserved in the context-specific profile because the proline within a structural domain is often useful or necessary for making a turn. And now let's look at the performance of this method. So we used a standard scope benchmark with a maximum pairwise identity of 20% to eliminate the trivial cases of similarity. And we defined everything as a true homolog, true positive, that is in the same superfamily, and everything in different folds to be a false positive, with a few well-known exceptions of interfold similarities, like beta propellers. And you see that here the BLAST curve is here. So this is a logarithmic scale. This was proposed by Kevin Karplas. We happily took up this this hint. And you see that BLAST finds about, or CS BLAST, the red line, finds about a factor of 2 or almost 2.5 more homologs at the same false discovery rates as BLAST. If we wanted also to show that this method works also in an iterative way. So I'll just remind you of how PSI-BLAST works. PSI-BLAST takes the sequences from the BLAST search, and the ones that are significant with an e-value below 10 to the minus 3, builds an alignment out of them, and from this alignment it calculates a sequence profile. And by now you know, of course, that if you have more information about each of these residues, namely what kinds of mutations you've observed in homologous proteins, you have more information now to search for further homologs. So PSI-BLAST is a very powerful method. In the second iteration, it often finds many more homologs than BLAST could find in the first iteration. Now, the simple extension that we do is that instead of adding pseudocounts to the alignment with the standard substitution matrix method that PSI-BLAST uses, we use these context-specific pseudocounts also in this step, not only here. And so then we can iterate, and we call this context-specific iterative BLAST. And when we compare this to PSI-BLAST, this is the first iteration again, in a different scale. This is the second iteration of PSI-BLAST and the second iteration of XI-BLAST. We see that still we gain quite a lot. It's not as much as BLAST, but it's surprisingly much, because remember that now we have, in the second round, we already have a family-specific representation of our sequence. So we have a lot of information that is family-specific. And still we gain a lot of sensitivity by including the horizontal information in addition to the vertical information in the alignment. So the information, even after the second iteration, is partly orthogonal. And then when we look at the fifth PSI-BLAST iteration, it is comparable to the second PSI-BLAST iteration in the lower selectivity range and the higher selectivity range. Two PSI-BLAST iterations are still much better. And then if you do a third iteration of PSI-BLAST, you gain everywhere, still quite considerably. This is also reflected in the alignment quality. So if you look at how many residues you can, of the alignable residues you really find, you really align correctly in the two methods, depending on sequence identity here, you find that CS-BLAST outperforms BLAST everywhere by some margin. And the same is seen in alignment precision. So how many of the residues that you actually aligned are correctly aligned? There you also see an advantage of CS-BLAST, in particular at the most difficult cases. Now, we showed in this paper that we have still a little problem or drawback with respect to BLAST, and that is the e-values. The e-value calculation is just used from BLAST. We don't change BLAST in any way. We just call it. We found that we have a problem here. So this is the reported e-value in this large benchmark all against all comparison, and that's the actual e-value. So at, for example, an e-value of 10 to minus 2, we should observe a certain number of proteins, of false positives. We observe a factor of 3 or so too many here. So we should be on this diagonal line, but we are in fact a bit off here. And the main reason for this is repeat proteins. And this is easy to understand because the null model that underlies the calculation of the BLAST e-values is a random sequence model. But if you search with a repeat protein, in fact you find other repeat proteins as high-scoring false positives. And these repeat proteins do not obey the random sequence model since they are repeats. So in other words, if you have, for example, a repeat protein and the one repeat aligns to another non-homologous repeat with just a little, a score of 5 or so, which is not significant by itself at all. If you now have 10 of these alignments strung together, you will get a score of 50, and that looks highly significant to BLAST, or to CS-BLAST in this case. So we looked at this problem, and we were able to solve it by including implicitly a repeat detection in CS-BLAST, which doesn't increase the calculation time. So the whole the generation of this profile takes only a second, which is negligible compared to the 1 minute or so that BLAST takes. And this repeat detection doesn't change this, doesn't make this any slower. And with this correction for repeat, so it's an e-value correction for repeat proteins built in, we get an e-value estimation that is really similar to BLAST, and we eliminate these bad cases of high-scoring false positives for repeat proteins. Now, summary, this is not the last slide, so I want to warn you, there are 3 other ones afterwards. The summary, first of all, we think that sequence searching still is a very interesting topic, and sequence analysis, and that they are really important because they are so much at the basis of many methods that are downstream of sequence searching and alignment building. CS-BLAST finds twice as many remote homologs as BLAST and has better alignment quality at similar speed. And CS-BLAST with 2 iterations is as sensitive as PSI-BLAST and it's more sensitive in the high selectivity regime. What is interesting is that the CS-BLAST binary is just a wrapper around BLAST, as I mentioned. So you can use all the same parameters that you can use for BLAST. Nothing's changed except the output format. There we still have to extend to other output formats. So instead of calling BLAST with your usual parameters, you can call CS-BLAST. You only have to give it the profile library here and the BLAST path, and then the rest stays the same. The output obviously also looks exactly the same as in BLAST. And as an outlook, we think that this is a very general idea that can be applied everywhere almost everywhere in sequence analysis and sequence searching, so multiple sequence alignment, molecular evolution. And our next step is now to apply this to genomic alignments. to find functional regions through their conservation signature, and we are quite confident that we can improve alignment so that we have more sensitivity to find small conserved elements in a big sea of unconserved, neutrally evolving sequence. I want to also give you a bit higher level summary of this work, a very high level summary, so to speak. I think bioinformatics is now driven really by information. It's not driven by algorithms anymore. I mean, algorithms are still important for certain things, but information is really has really gained the top hand, and that was also what guided our work here. So, if you have a sequence you want to search with it, it was crucial at least for us to understand that there's some sequence, that there's some information that is not used, and that's the information about the context of every residue. And then once you know what the information is that is not used yet and that you could use for making predictions or understanding your system, you need a good statistical model. So statistics is, I think, the new ruler of bioinformatics, and the enormous success of machine learning methods, in particular those that use sound statistics, is, I think, a sign of this. Just to give you two other examples, we have developed a method called HHPRED, an HH-search that is able to find very remote homologs, and it can be used for structured prediction. That's very successful. That also relies on just taking more information to do predictions, to find remote homologs than has been done before. And another example from yesterday's keynote lecture, Trey Ideker, I think, has understood very early that in order to understand transcriptional networks, it helps a lot to pull in information from protein-protein interactions and use this information in the analysis. I want to thank in particular Andreas Biegert, with whom it is enormous fun to work and who will continue with this genomic alignment, and the rest of the group at the Gene Center Munich. And I want to point you to our web server where you can try this method. You can download the binaries for free for academics. It's for free, and you can also use the web server here. It's the same as BLAST. Same, you just paste your sequence, click on the database you want to search, submit the job. There's also now a web service, CS-BLAST web service available from EMBnet. We hope that others will be interested. NCBI is still testing. And there's a poster, U08, where Andreas Biegert will be after the session to answer questions that you might have. And there is a demo on this bioinformatics toolkit where CS-BLAST is integrated at 3:45 in room C8. Thank you very much.
Thank you very much. Questions, please? We have 3 or 4 minutes for questions.
Yes.
Hello, I have a question about what do you mean with homology? Because I think you can only get information about sequence similarity with BLAST, not directly about homology.
That is true. So, I mean, homology obviously means that you have a common ancestor. But a degree of similarity that is not expected by chance is a strong indicator of homology. And that's why BLAST and PSI-BLAST is successful. I mean, that's the whole logic. And we just use the same logic. It works very well.
So you mean, in principle, sequence similarity. You don't mean homology when you...
This is a method that uses sequence similarity, but it finds homologs.
So that means it's not only the sequence similarity, but also... there is something additional, so you get the homologs? Or?
No, I think it doesn't use homology in any other way than BLAST or PSI-BLAST does.
So it's sequence similarity?
Yes.
Okay,
yeah.
thanks.
Yeah, I was wondering whether it would be possible to also get to get the sequence-specific the context-specific profiles. And I was wondering, from a machine learning perspective, when you try and encode the protein sequence, sometimes you use a PSI-BLAST, but which of course takes time, and this would be much faster.
Yeah, that's right. So you after 1 second you can have your your context-specific profile much faster than a BLAST run. You can also, you could take this profile and do machine learning, apply machine learning techniques on it. You can also use the probability values that we calculate for the for all these clusters. So we have Andreas Biegert has written a binary that will put out this matrix of 4,000 times L numbers with all the posterior probabilities.
Okay, and that's included in the binary?
Yes.
Oh, thank you.
Hello. You mentioned problems from repeat regions, okay,
yes.
in the values. And I was wondering, how do you detect those regions, and where do you correct the values when do you exclude those proteins? Do you mask the proteins? Do you correct the value only and then exclude the results? What do you do exactly?
No, we don't mask anything, so we still find homologs for the repeat proteins. The only thing we do is we correct the null model, basically. It's a heuristic, and what we do is we give little penalties to the positions that look like repeat positions. So if we find a similarity within the protein, within itself, we give very, very small penalties of 0.1 bit or smaller. And these penalties are just enough, to suppress all these false homologs, because these false hits are no good hits. I mean, if you have 5 bits in an alignment of 30 residues, it's a very bad score. So if you subtract just a little bit per column, it's enough to suppress those false positives.
Yes, the problem is that Astral is very poor and SCOP in general is very poor in repeat regions and disorder regions, because
exactly.
You cannot crystallize them. So,
right.
Have you done any other tests to see how the method performs in real-life scenarios?
Right, that's a good question. That's how we have found out about the repeat problem. That we didn't find out in SCOP. We found out by applying this method, for example, to the human genome, and we found cases where the method took 10 times longer. That's in fact how we found it. And we looked at these cases and found that the reason was that we got huge lists of false positives for proteins with very long repeats.
Okay, thank you very much.
Okay, so there are many more questions I see. However, we have to cut the session now, and cut the talk now, and switch to the other talk. So let's thank the speaker again.